Okay, guys, we are going to get ready. Maybe now it's on. Hopefully people online can
hear me. If they can't hear me, then so be it. Thanks for joining us for this last workshop of today. We're going to be going over Unraveling Immunogenomic Diversity in Single-Cell Data with Ahmed Al-Jami. And he'll take you through how to access all the tutorial servers. And he's pretty much self-sufficient, so he doesn't need anything more from me. Take it away.
All right. Well, thank you very much for the introduction. My name is Ahmed. I'm a third-year PhD student from Lebanon. I'm doing my PhD actually at the University Hospital of Goethe University in Frankfurt, Germany. And today I'm going to explain to you how you can unravel immunogenomic diversity in single-cell data, or maybe attempt to explain. So let's see. So first, it is important or essential to understand that structural diversity of receptors is very important for to coordinate antigen-specific immune responses. We can see here T-cell receptor, or TCR for short, that is recognizing an antigen being presented by either an MHC class 1 or class 2 molecule. This triggers an immune. Triggers an immune response, or followed by an immune response, as illustrated here. What I'm showing here is a single MHC class, like a single MHC molecule or HLA molecule with one particular structure. But it is important to realize that this structure needs to be diverse to be able to recognize a wide variety of antigens. So to ensure this structural diversity of these receptors and molecules, we have different genetic mechanisms, and the focus today is going to be on two of them. The first one is about polygyny, where you have multiple similar genes encoding same or similar function. This is the case in MHC class 2 molecules, where you have different genes encoding different proteins or molecules, which pretty much have the same function or similar function. This is also the case in immunoglobulins, where you have different constant segments for the light antibody chain. They all fulfill the same function, but they just have the sequence slightly different. The number of those similar genes in such gene groups is relatively low and easy to handle in this case. The other one is about hyperpolymorphism, where you have multiple alleles for one gene. And we can see here, this is a representation of diversity on a sequence-based level in MHC class 1 and class 2 molecules. And the red color here represents different substitutions in the nucleotide sequence that leads to amino acid exit changes. We can see that this red color is actually present in the antigen binding cleft, which further highlights that these structures need to be diverse to be able to recognize a wide variety of antigens. The number of alleles in such gene groups here becomes much larger, and this creates different bioinformatic challenges. I think we've heard enough over the last 2 days about single-cell experiment and how to analyze single-cell RNA sequencing data. So in a typical transcriptomic analysis workflow, this gets complicated, particularly at the step when we have to map reads to reference genome. Because this pretty much creates a bioinformatic challenge. In this case, it's called cross-mapping, where we have reads uniquely mapping, but unfortunately to the wrong gene. So what we want to do here is to perform quantification of allele-specific expression of immunogenomic analysis. So what do we want to get? So what we want to get eventually is to build a multi-layered data structure for immune gene representation in single-cell data. And I'm just showing here like a brief introduction to the demo, but this will be much clearer once we step into once we start coding together and step into the demo session. But just to continue, so here we have two different genes. We have the gene A1 and gene A2, and these two genes encode the same protein subunit and have many different alleles. So these fit what we tried to talk about. They are like a representation of genes that are hyperpolymorphic and polygenic. You can see from different donors, we get different alleles for those two genes. So what we want to build from that is a multi-layered data structure, as I mentioned, The first one would be on allele level, where we have cells in columns, so alleles in rows. The gene level is pretty much what we have nowadays, or like with a single cell experiment, where you have genes on the as rows and cells as columns. And the last one, functional, which we will also be clear once we start coding, is that when we start pretty much aggregating the alleles to get one particular function. This is the case, as I mentioned, in immunoglobulins, where you don't really care which of the constant region, which one of the constant genes the antibody light chain is made up of, but you only care that the function is there and it's working just fine. So how do we get to all of this? In order to unlock deeper insights from single cell studies and to overcome the bioinformatic challenges that I tried to talk about, I present you here a workflow that represents or that comprises 2 software packages and 1 data package. The first software package is called scIGD, and this is actually a Snakemake workflow, so it has nothing to do with bioconductor. But this Snakemake workflow allows you to perform 2 things. First, you are able to perform allele-typing of structures, especially like HLAs. So pretty much you start being able to get okay, this patient or donor or whatever has these particular alleles, HLA alleles. This is the first thing. The second thing, it will allow you to perform allele-specific quantifications of genes and those typed alleles. So, and pretty much that's what we get. So you have different alleles. You get exactly what you get with Cell Ranger or with Callisto or any of those tools. You get a count matrix, features in rows, cells in columns. And in this case, instead of having only genes, you start also to see the alleles. So what this requires is 2 inputs, and I will show you in a minute. 2 things. So gzip row FASTQ files, and then the reference genome as FASTA. After that, after having this count matrix, then now you can jump into feeding this count matrix into a data structure that we develop, which sounds familiar. It's called single cell allele experiment, since it's pretty much an extension of single cell experiment objects. But it offers you this multi-layer data structure that we're going to talk about. This also allows you to perform your downstream analysis on any of the levels that I tried to show here, on a gene level, allele level, and functional level. Single cell allele experiment is now a bioconductor package since last release. And example data sets, just to show you how the output of scIGD might look like. We have it in SCAE data, which is also now an R and experiment hub package since last release. So to start, I'm gonna briefly walk you through first the 3 tools that I just showed you here: the scIGD, the single cell allele experiment, and the SCAE data. And then later on, of course we will go together in the code. But just to spend like a few minutes on the Snakemake workflow. This is scIGD. It's so it's on GitHub now, and pretty much we have an extensive documentation in this section. This works for 2 at the moment 2 technologies. It works with 10x version 1, 2, or 3, and also with BD Rhapsody. And in BD Rhapsody, it works with either amplicon-based or whole transcriptome datasets. Yes, please.
Do you need any particular sequencing length for this to be effective or non-effective?
No. Of course, if the sequence is long enough, you don't need any of this. But so, because then you don't have this cross-mapping issues and other issues if the sequence is long enough. But if the sequence is not long enough, then you need this. And this has been tested on, let's say, 70 base pairs, around 70 base pairs and stuff, and it works pretty well. Yeah, but it's also been tested, as I said, on 2 technologies, the 10x and BD Rhapsody. And in BD Rhapsody, it's been tested also on amplicon-based and whole transcriptome, and also on multiple donors. And the idea behind doing the allele typing is that you're using, to avoid all of those bioinformatic challenges and cross-mapping, you're actually using donor-specific reference genome. So it's not like one reference genome that is being used everywhere. It's donor-specific reference genome where we get those alleles from. As I said, I'm not going to spend too much time on it because it's out of the scope of Bioconductor. It's Snakemake. But as I said, the documentation is right here. It takes you from what you need as a first you can install it through Conda or Mamba. You can create the environment there, and then what kind of input data and in what kind of format and style they need to be. And as I said, it's pretty much gzip fastq so raw data and the reference like a FASTA file. And of course, if you have multiple donors, then it expects to get that you give it a sample tag sequence. Then so it does the demultiplexing for you, because that's also a part of the workflow. And then, of course, as a Snakemake workflow, it has a configuration file. The documentation tells you what kind of parameters are there, how you can change them, and what you need to put. Then you can run the workflow in pretty much one line. And that's it. You can specify your memory and cores and data allocation. And the output, last but not least, the output is going to be 4 files from scIGD. It's a Callisto output, since that's what we were using for the quantification. So that means it has a matrix, like a count matrix. It has a text file, which is the features, and it has a text file, which is which has the barcodes, and a lookup table for the HLAs, and that I will show you in the demo. This just tells you which allele or which gene this allele belongs to, and which functional group or class this belongs to. So let's say we run scIGD. Now we have those 4 files. So what are we going to do with them? We're going to jump now into the demo, to the code. So first, and maybe I just say it out there. So the SCA data, as I said, is on Bioconductor now. The single cell allele experiment is also up on Bioconductor. This is for the workflow demo, and I will put the QR code at the end of the session. It has the slides, it has the code, it has all the materials that I'm showing today. There's a GitHub page if you don't want to follow and run the code today line by line. You can follow us through the vignette, which is also here. If you don't want to run the code, this is pretty much what we're going to run today, because I don't think we're going to be able to accommodate everybody on Galaxy. So if you want to follow the vignette, feel free. But if you want to follow us, then please hop on to Galaxy and load scIGD, which is this one. And just by a show of hands, I assume, is anybody not familiar with single cell experiment here? Perfect. All right. Then please run the tool, and it also, the link is also here for the material, actually. So I guess I'm hoping everybody's in. I already loaded this, or did I not? Did not. All right. Anybody? Anyone has an idea here? Otherwise, I would run it from my local R. But anyone has an idea?
Click on this again.
Just new one,
And then go for advanced.
advanced.
Sorry. Yep.
Go for advanced and use certificate.
View certificate.
Scroll down.
Scroll down and take the risk.
Yeah, take the risk.
Can
you go all the way down?
That's all the way down.
Oh, what about
Previous tab. Go back one.
Go back to
Usually there is like they said, like a risk.
learn more.
Yes, try to learn more.
Not more.
Yes. No, never mind.
All right.
Try with Chrome.
Yeah,
Should give you the option to
Yes.
take the
let's
risk.
see, let's
Chrome
go.
will let you get hacked. It's okay. Use that.
What's that?
Nothing.
I need my password.
Show your last one.
What's the new one?
Yeah, I'll let one of them.
Okay. Sorry for that. We're back on. Let's do it now. Yeah. Where is that? Thank you, Alex. All right. Hoping everybody's in. Whoever wants to be in is in. So, yeah, so we talked about the overview, the description. I mentioned scIGD, single cell allele experiment, SCA data. And now we're going to jump into loading the libraries first. And it might take some time so we can talk in between, while the commands are running. So first we start by loading some packages. Of course, we need single cell allele experiment. We need SCA data. SCA data you typically don't need, like if you want to run your own data, but this is just to show you to work on one example data set and showcase what the output might look like. So what is SCA data? Just if you want to look at SCA data consists of 3 single cell allele experiment. I run this in between. Data sets that were sourced from publicly available data sets provided by 10x Genomics. We have 3 data sets, PBMC 5K, 10K, and 20K. And these were all taken from the 10x Genomics website, thrown into scIGD. And then from scIGD, we got the 4 files that we're talking about. And since this is an experiment hub package that we're trying to load, so we need to make sure that this is saved somewhere. So it's going to ask you if you want to create a directory. So we say yes. And it's one command or one function. It's called SCA data get. You specify the name of the data set. You can say PBMC 20K. You can say PBMC 10K, or you can say PBMC 5K to load one particular data set. And what this is going to give you now is, as I mentioned, because it's on Experiment Hub, so it will give you the directory element, which is specifying the directory where files downloaded from Experiment Hub are stored on your device. And the 3 remaining elements are going to be the barcode, the text file, the features text file, and the matrix, the count matrix text, matrix. And so this is how it looks like. It's just the directory of these. So as I said, the fourth file that we get from scIGD is a lookup table. A lookup table here corresponding to each data set. So it facilitates the creation of the relevant additional data layers during the object generation of the single cell allele experiment object. And the lookup table looks something like this. So for those who are not familiar with HLA or MHC molecules, in this scope, we're working with 2 types of HLAs. We have HLA class 1, which comprises HLA-A, HLA-B, and HLA-C. And if we want to look at and there's also HLA class 2, which HLA class 2, which has HLA stuff like DPB1, DQA, DQB, and others. And this is how the lookup table looks like. It tells you what kind of alleles you have for the DPB1, for example, and to which function it belongs to. This will be very important for the creation of the multi-layer data object. And you might probably know this, but at most you can have only 2 alleles per gene. So
these alleles are sequencing variants, not like isoforms of not differential splicing. It's SNPs or whatever they are.
Mm-hmm. So I'm just gonna run this while I explain. So now we have the data we load, and we want to create we want to create a single cell allele experiment object out of it. So the read-in function, this is very important for single cell allele experiment. It's called read allele counts, and this is used to read in the data and generate an SCAE object. So when using your own data generated by scIGD, so of course we need to state the path containing all the expected files to the parameter called sample directory. And this is the 3 files that I mentioned, where the matrix is, where the barcodes and the features. Then you need to specify to the parameter lookup table where the lookup table is. In case you rename the files, then you also need to mention the rename of those 3 files in those parameters. Otherwise, you can ignore those 3 parameters and just put the directory where you have all 4 files in the sample directory. Then you have 2 options for filter mode. You can or 3 options actually. You can specify filter mode to no, and this is the default value. This will perform filtering based on a threshold 0. So filtering out columns, aka cells, with a count sum of 0 over all rows, aka features. If you want to do a bit of filtering here, that and then you don't have to worry about filtering, at least for one particular type of cells, then you can specify filter mode to be yes. And this will perform advanced filtering on the computed inflection point of a knee plot based on a barcode ranks. So the information to the corresponding plot, including the inflection point and so on, will be stored in the metadata of this SCAE and then knee info. And of course, if you are into filtering and you want to give a custom threshold for filter mode, then you can, after looking, of course, on the plot and the inflection points and so on, then you can specify this threshold here. So you say first filter mode custom, and then in filter threshold, you specify the threshold that you want the cells to be filtered on. Then you have also here the option to normalize the data already, so you don't have to do log whatsoever on your own. And this is actually, we recommend, unless you're very into statistics and mathematics and better than we are, to keep it true, because this will perform normalization on the row counts, which is the default value. Please note that this count matrix is extended. So the initial count matrix is pretty much features plus HLA alleles. We don't have just yet the HLA genes and the HLA functions. These will be built from the lookup table. So the count matrix is extended. So we're adding actually additional rows during the object generation. So it is crucial to compute the size factors only on data layers present in the row data. So the non-immune features and the HLA alleles. Otherwise, the immune features would contribute to the size factors multiple times, and thus the size factors would be incorrect. So this is advised to be log equal true. And then you also have, since it's we're getting from scIGD ensemble entries, then you can also specify here in this parameter gene symbols to be true, and this will add the corresponding gene symbols of the ensemble IDs to row data. This is how the function looks like. So you call it SCAE. You do read allele counts. The sample directory is the data $ directory where it is. This you don't need to mention when you're working with your own data, but here we mention it. Where the lookup file is, so lookup that we got from here, so the read CSV file. And then you can say filter mode yes, log true, gene symbols true. You can give your sample names a name, and this will be added as a column data. And then, yeah, to see it. So, and this again, I say it, when using your own data generated by scIGD, you only have to populate samples directory with the folder path. All the others can be left untouched, like those three. Of course, unless you change the names, the naming of those files that is coming from scIGD, then of course you need to provide this. So it tells you what it's doing. It's filtering. So it starts filtering filtering performed based on the inflection point. It says that it's using this package to retrieve the NCBI gene identifiers. It tells you that it's extending now row data with new classifiers, filtering at this level of UMI count. It's computing library factors before adding new layers. It's now aggregating alleles corresponding to the same gene. So how we do this is pretty much just a summation. Now you have, let's say, two HLA alleles. So you sum up those, and then you get the gene level. And then we're aggregating genes corresponding to the same functional groups. So this is, let's say, HLA class 1 has, let's say, HLA A, B, and C. So you're aggregating all those three to get like a count sum of the functional group. And then we're doing normalization. And then this is exactly what you have. This looks very familiar to everybody. You have now the class single cell allele experiment. You have the number of rows, columns. You have the knee info, as I mentioned, to look at the knee and inflection point values. You have the counts, log counts, the identifiers, and some other row data that we're going to talk about now or soon, the barcodes. And what's different from single cell experiment is just the last lines here. So it tells you how many immune-related features. So these are the HLA. It has 23 of them. Allele level information, it has 14, and it mentions which 14 those are. The 7 immune genes, so the HLA A, B, all the way, and the 2 functional groups that we have here, HLA class 1 and the HLA class 2. So let's showcase the content of this of these object slots. So 2 new classification columns are introduced here in the row data slot. 2 of them are NI NII column. So this is a classification of each row as non-immune or immune. Immune meaning it's the HLA features. And this will be useful for how to, let's say, navigate through all of those layers, how to subtract or subset, sorry, the allele information, the gene information, or the functional information. And you also here have another row data, which is the quant type column, which specifies that this row is either an allele, a gene, or a functional class. So, and both columns are used jointly to match each row in the object to its corresponding data layer. And it looks like this. This is the row data of SCAE. So it's the ensemble entries, and then the alleles, the genes, and the functional groups. So NI here meaning non-immune, so has nothing to do with HLA. These are immune. And the quant type, so the quantification type, G is gene, and you have the F, and you have also the A for the alleles. And then the NCBI ID here at the end, if you ask for it. So then to look at the column data, this contains information about the sample and barcode information and the size factor if you have asked for log to be true. So how do we extract layer-specific figures now? So there are getters that are implemented to retrieve the different data layers integrated within the SCAE object. So to start, if you just want to ignore all HLAs and want to work with all the non-immune features, you can use a function here called SCAE subset, and then you can specify on which subset you want. And in this case, you can specify non-immune. And then as you can see here now, it has a total of 0 immune-related features, 0 alleles, 0 immune genes, and 0 functional level groups. If I want to extract only the alleles, you can specify it like this. And this will, so you mentioned alleles here, and then you see here your rows are only 14, and you have all the alleles here. For immune genes or the genes now of those alleles, you can mention immune genes, and you can go on and on. But of course, you're not gonna just, let's say, get a subset of 7 genes or something. So you're using this in combination, as we're gonna see in a few minutes in the downstream analysis. And these are the functional groups. So you specify these functional groups, and then you get only 2 rows, which are HLA class 1 and class 2. So to look at to perform some downstream analysis, let's start with pre-processing workflow. In this case, we're just removing features that have a total count less than 5. So with the exception of HLA immune features. So you're just gonna do this. So here we're specifying NI, so we're extracting the row data where NI is I, so this means immune features, so alleles, genes, and functional groups, and yeah, and it looks like this. This is gonna be how it looks. And now we can extract the other genes that are not, so this is just a negation, so not immune features. I really like to use it like this. I don't know. Does anybody use it like that?
No.
It's really cool.
Gotta do the exclamation point.
No, this is really cool. So you just negate on that. All right. So now we're gonna remove all features that are less than 5 while keeping all the immune features. Is anybody running code actually? Yeah. Okay. If I'm going too fast or too slow, let me know. If you need more time, also.
Yeah.
Yes.
Also, that Alex scaled up quite a bit if you wanted to join in.
Yes, please.
Although the point of the demo is to not wait for anybody, because that's kind of the point of the demo.
Okay.
But it's here that anybody can go back later
Sure.
and do it as
Exactly.
slowly as they want.
Exactly. So we removed now some features. We're left with this many features and this many cells. So let's start by doing some processing work, like processing the data. So we start with normalization. As I said, if you specify above the log equal true in the object creation, then you don't really need to do anything here. So the data has already been normalized upon creating the single cell allele experiment object. So now let's before we start doing the downstream analysis, let's just look at the HLA immune features to see how they are. So we're just going to plot their distribution. second. So while it's running, this is pretty much the kind of image that we're gonna get. And okay, there it is. But anyway, it's also here. So we have all those alleles, like the A alleles, the B, the C, and all of those. You can see the expression in log counts here. You also have the genes now, and you also have the functional groups. And of course, the functional group again is an aggregation of the genes. So that's why it has the highest expression level. So this just shows you first that we were able to perform allele typing well, and we were able also to do allele-specific quantification, as we can see in this kind of results.
A question about input data.
Yes.
So you mentioned like references, patient-specific references.
Yeah.
So do you have to do like genome sequencing before that? Or?
No. As I said, the input. I can go back to it quickly. The input is pretty much like this. So you need your gzip row gzip fastq files. This you have from 10x, from BD Rhapsody, whatever it is, and you need this. So the DNA primary assembly, the FASTA file, and the GTF file, and these you can install from Ensemble. So like.
Yeah, okay. I mean that the allele alignment is done
with the scIGD. Yeah.
And kind of tries to infer the alleles that are present.
Yes, exactly. Yeah.
And how it could, because some of them are like similar to each other.
Yeah.
Still have some kind of uncertainty, or because here you just have some particular set of the final ones.
Yeah. So yes, you're right. The sequence is usually very similar. There are different tools to perform the allele typing. The one that we're using in the Snakemake workflow is called ARCAS HLA, which has been benchmarked even by 10x. 10x tried to develop their own tool to do allele typing. It was benchmarked to ARCAS HLA, and ARCAS HLA seemed to perform best when doing the allele typing. Yeah, they are very, and the way they do it, like ARCAS HLA is using. So they have like the allele database that they try to align to, and then they use their own model to get like those two alleles.
How do you deal when there are like sometimes there's multiple typing result possibility? They give you more than two possibilities.
So they give you they report different percentages, like by confidence, which two are like got the best result from the model. So they give you a list of many alleles, but then they choose at the end the one with the highest percentage. This is what you get as the two alleles. Sometimes it's one allele, but they do maximum. Yeah, so they have the model where then they try to align to so many different alleles, and then the ones with the highest percentage that fit the model best, this is what's reported. All right. So before I talk, I will run this because this will take some time. Yeah, so this is it. So now we're switching to the downstream analysis, and this is the section that we want to focus on for the rest of the time. So in this section, we're going to provide insights into the various data layers of the SCA object and offer guidance on performing HLA immune gene expression analysis. So what you're used to, PCA, UMAP, Disney, building the SNN graph and performing clustering, this with single cell allele experiment can be done in three different ways. You can perform it on either so you can perform it in three different ways depending on the question that you're trying to answer, depending on the analysis that you're trying to perform. Each has its own pros, and as I said, depends on the question that you're trying to ask. So you can do it on the non-immune genes, on a combination of the non-immune genes and the immune genes. So this is pretty much like single cell experiment, how it's implemented. But you can also do it on non-immune genes plus immune alleles. This might be beneficial, for example, when you're trying to, for example, you have different clusters where you have genes being expressed on similar levels in the clusters, but then the alleles are the ones that are differentially expressed. So this could be helpful to see from here. Here, you can perform on the non-immune genes plus the immune alleles. This could be helpful, for example, when you're trying to, when you have different clusters of the same parent cell type, but then you have, in the same cell type, you have different differentially expressed alleles. And the last one is that when you really don't care which HLA class or HLA class 2, or when you have multiple donors and you don't really care which one of the HLA genes is there or the allele, then you can perform it on the functional group just to make sure that it's working, like the function is there and present. So this will allow us the representation of immune genes at various levels, from alleles to genes to functionally similar groups. And this will enable the user to perform these kind of comprehensive analysis across the different layers, enabling different angles on the same data set at hand. It is important to note and to create unique identifiers for each layer or run, otherwise the results will be overwritten and saved incorrectly. So imagine if you run PCA 3 times on all, so by default, these will all be saved to PCA, the name PCA, and they will be overwritten. But if you want to have your own, if you want to keep, let's say, track of those 3 layers, so then you have to specify the name of PCA, as I'm going to show you now. For simplicity here, we're performing dimensionality reduction clustering on the gene level, using the immune and non-immune genes after choosing only top 2% highly variable genes. We do it here only for memory reasons. So you start by extracting the genes, you then run this, the model gene variable, then you choose the get top highly variable genes, you specify the proportion, and yes, so this you don't have to do. You don't have to subset SCAE to keep this, that we're doing here for memory reasons. And then you run PCA, for example, on this chosen HVGs. And this is what I meant here. It's important that if you're running it on genes to name it like _G or something. Otherwise, if you attempt running it on alleles next, it will be overwritten. Then you can use this to run UMAP or to run TISNE. You can build the SNN graph with whatever K parameter you want. Then you can use something like iGraph or Louvain clustering to get the clusters. And then you can look at the UMAP and TISNE. And we do this here. And this is, at least for this data set, we get something like this, 8 clusters. And yeah, so then you can start with visualizing the allele on those clusters. And this is running at the moment. Maybe one note for those who are also not familiar with HLAs. So HLA is, so there are 2 classes, as I mentioned, class 1 and class 2. Class 1 is pretty much present on almost all nucleated cells. So you can find it almost everywhere on all cell types. HLA class 2 is more on the antigen presenting cells, so like something like dendritic cells, macrophages, and B cells. So that's why you might not be able to see it on other cell types. This is what we're actually gonna see soon. So while this is running, I will just jump to the next part. If you want to visualize those HLA and immune features, so here I'm showing you an example of a gene and its alleles. So 1 gene, 2 alleles. And this type of representation will help you determine which of the 2 alleles has a greater influence on the gene's expression. So we open this on TISNE. You have this HLA DQA1. If you look at it with single cell experiment as it is now, so this is pretty much all you will see. So it's expressed in these regions and so on. But if you look at the 2 alleles, then you can say like, okay, well, this patient or donor has these 2 alleles, but one of them is actually has a greater influence and is dominating the gene expression level. So you would know exactly, let's say, which one of the 2 alleles is giving or providing this gene expression level. And you can do it, yeah, with a plot reduced dim function. I think this is from Skater. Yes. Yeah. And maybe this is running. This one takes a bit of time. Just gonna jump to TISNE. Again, we can use something like plot reduced DIMs if we want to look at all the different immune features in the clusters, and to be able to see like, okay, well, which cluster or region this allele or gene or functional class is most expressed in. So we can do something like this. And then we end up with a figure like this, where you have, I'm just showing you here the 8 clusters on t-SNE that we saw previously, and then all the immune features next to it, just to see like where are they present and in which clusters exactly you have the expression of those. And this has all the, and HLA class 1, you can see now it's everywhere, but HLA class 2, as I said, HLA class 1 is most on almost all nucleated cells. HLA class 2 is on antigen presenting cells. And you can see that it's not here. All right. And I have a couple more figures to show here. We can also use violin plots. And the violin plots in this case are allowing us to identify differences in allele expression within each cluster. So imagine now, okay, I have those 8 clusters. I want to see like the differences in one gene's allele expression difference in the cluster. And you can see here, for example, on cluster 3 or 4 or whatever, and here are the names. Then you can start saying, okay, well, at least this cluster seems to have a lot more of this allele than the other allele, and so on. So this provides you with this kind of information. And then we can also look at something like a heat map, and heat maps in this case will enable us, the way it's written, at least in this chunk, will enable us to identify upregulated or downregulated alleles across all the clusters that we have. So it's going to look something like this. Then you can also start identifying differentially expressed alleles and so on within the, like, across all clusters. So you can see here that this DQA1, for example, is highly upregulated in cluster 6 compared to the other clusters. And I think this is last figure here. We also have bar plots. So the bar plots in this case will allow us, in how it's written again here, will allow us to compare allele frequencies. So we're looking here on HLA class 1, meaning HLA-A, HLA-B, and HLA-C, and the cluster number here. And you can see on the y-axis here the total allele count, so the log counts. And then you can also start identifying, for example, if you look at the cyan, I think, or turquoise color here, this is the B44 allele. You can see that in almost, actually in all clusters, it's more expressed than the other allele, like B15, and so on. So you can start identifying which allele is actually dominating the expression level. We can do the same thing on HLA class 2. And this is how it might look like in HLA class 2. DQA1, 2, 1, for example, this has much more greater influence than the DQA1. And same for the orange compared to the red DRB1 alleles. I hope that's making sense. In case you're still running, you can run. Last section that I want to mention is that if you want to perform interactive exploration of single-cell allele experiment objects, you can use it with IC Shiny app to perform analysis, because it's simply, since SCAE is an extension of SCE objects, so it's also compatible with IC package, and it could be as simple as running IC and then SCAE. This will start a Shiny app where you can analyze your data, look at your data the way you want. And you can also here, if you're creating different t-SNEs, different UMaps, you can view all of those on this Shiny app. So you're looking, again, same data, but from different angles. So that concludes the presentation. All the demo material, as I mentioned, including all the slides, the code, the links, everything is in this repo, scIGD workflow demo BioC 2024, and this is the QR code if people are interested to use the tools. I hope I convinced you, and if not, that's okay. And I just want to acknowledge our project partner, Federico Marini, for all of his valuable input, my PI, Katarina Imkele, and Jonas Schuck, another PhD student in our group, for all their input and hard work on this project. The GitHub repo for the 3 tools that I talked about today are also here. And of course, I also want to thank our funders and whoever is having us have bread on our table, Deutsche Krebshilfe and CZI, especially for the generous funding. Last but not least, thank you so much for your attention, and thanks to Bioconductor for giving me the platform to present my work. Thank you so much.
Can you go back one slide just so I can? Can you go back one slide just so I can? Yeah, thank you.
Are there any questions, comments? I didn't really discuss the biology, for example. I really showcased you the tools, but not much biology, or like how could this be applicable for time reasons. But one quick example you could think of is HLA loss, especially in tumor cells and tumor samples. Imagine that. So this tool would allow you to see like which of the HLA the tumor cell, for example, has lost. So this could be the case if you have a tumor data set. You can load this into here. Then you can compare immune cells against tumor cells. In an ideal world, or you would see both alleles in tumor cell and immune cells. But in HLA loss examples, you would see two alleles of one gene on an immune cell, but you would see only one allele on a tumor cell. This is in biology referred to as HLA loss, or tumor evasion. So the tumor gets smarter and loses one of its alleles, and this pretty much diminishes the repertoire of genes that this tumor cell could present to an antigen-presenting cell, or to an immune cell like T cells. So this is just one example how this could be applicable, among others.
I think it's a very interesting talk, and I have questions. But we are about to lose the room because the day is over here. But we will be back tomorrow, 7:30 for breakfast, 8 a.m. for our keynote. But I do want to hit you up because I have some really interesting concepts around like T-cell leukemias and stuff, where this could be really useful for tracking clone types.
Please do. I'll be very happy to answer any questions. I'll be here all like tonight and tomorrow. Please hit me up. My email is also here, and you can also open any kind of issues on GitHub. I'm very happy to get into